Okay guys, now we have to discuss reproduction, that is the first unit of 12th. In reproduction you have general account of reproduction in organism, that you have already discussed how reproduction occurs in Monera, Protista, fungi, Plantae and Animalia while discussing the diversity of life forms. And then second chapter, that is the reproduction in flowering plant. Third is reproduction in animals. So here we will talk about plant reproduction in flowering plants, because the first chapter, that is very general account, that we have already discussed while discussing the diversity of life forms. Now reproduction in flowering plant, this is the topic we have to discuss. Reproduction in flowering plants, that is anthophyta or angiosperms. So in case of angiosperm, for the reproduction act, we have to discuss about megasporogenesis, microsporogenesis, pollination, fertilization, embryo development, endosperm development, polyembryony, apomixis, parthenocarpy, and finally the structure of seed. So if flower is a reproductive part, flower is modified shoot. What is modified shoot? Flower has two types of parts. The one is reproductive part, second is non-reproductive part, right? So essential parts and non-essential parts. Essential and non-essential parts. Essential are the androecium and gynoecium, and here is calyx and corolla. Now you will find, first of all we will talk about androecium structure, then gynoecium, and finally how the pollen grains are produced and their gametophyte. If you talk about the male reproductive structure, that is androecium, and the unit of androecium is stamen. Stamen, you will see the structure will be anther and filament. Both are joined by connective tissue. Anther is a fertile part. अगर हम उसका transverse section काट दें तो you will find inside this anther there will be two anther lobes. This is one lobe, this is another lobe, and here is the connective tissue. In each anther lobe there is one microsporangium. There is two microsporangium in each anther lobe. So here this is the TS of anther. TS of anther. Here will be the anther wall. From this TS of anther we can say most of the anthers are bilobed, lobe दो हैं, and tetrasporangiate. Anther wall, you will find outside anther wall, outermost layer will be epidermis, that is protective, then endothecium, middle layer, and tapetum. Tapetum is a nutritive tissue that will provide all the nutrient for the developing pollen grains. Now, this microsporangium, इसके अंदर की जो cells हैं, they are the microspore mother cells. These microspore mother cells, आप इन्हीं को pollen mother cells बोल सकते हो, या you can say myocytes, जिसमें meiosis हो, they are called myocytes. These myocytes will undergo meiosis, and myocytes will undergo meiosis. Myocytes reduction division, and formation of microspore tetrad. Microspore tetrad. They will be the microspore tetrad. Phenomenon will be called microsporogenesis. Microsporogenesis. अब ये haploid microspore बन गए. Now these microspores, this common wall, that is tetrad की common wall made up of callose. Callose wall will be broken by callase enzyme, and all the microspores will be separated from each other. And all the microspores जब एक दूसरे से separate हो गए तो they can be said as pollen grains. Now these microspores or pollens will undergo microgametogenesis by the act of mitosis. First division mitosis का asymmetrical, एक larger cell बनेगा, that is vegetative cell या tube cell, and second is generative cell, that is the smaller cell. Gradually generative cell will construct and will get embedded in the cytoplasm of vegetative cell, and vegetative cell will also get surrounded by the outer exine wall. This is the exine, that is made up of sporopollenin. This sporopollenin is coming from tapetum, and here is intine, that is made up of pectin and cellulose. So this is pectocellulosic. Now this pollen grain from the germ pore area, this vegetative cell will elongate, producing the pollen tube, and generative cell will enter into this growing tube. Finally the pollen grain will be fully developed, and fully developed pollen grain generative cell में एक mitosis हो जाएगा, तो 2 male gametes will be produced. Growth of the pollen tube occurs from the tip portion. To prevent the cytoplasm at the tip portion of growing tube, there is formation of callose plug. Here is 2 male gametes, 2 sperms. They will be used in the act of double fertilization. This is the vegetative nucleus. This whole process is microgametogenesis. This is the structure of male reproductive structure. तो pollen grain release from the microsporangium either at the 2 celled stage, या तो 2 celled stage में आएगा pollination के लिए. 2 celled stage होगा तो 1 will be the vegetative cell and 1 will be the generative cell, or in some plant it will be at the 3 celled stage. 3 celled stage में generative cell has taken one more mitosis, जिससे 2 sperm का formation हो चुका होगा. Now structure of pistil, you will find pistil, that is the collective term is gynoecium. Gynoecium की unit that will be called carpel or pistil. Carpel or pistil. The structure here you will see, this structure of pistil will be like this. The elongated structure, this is style. The uppermost part is stigma, lower part is ovary. Inside this ovary you will find presence of ovules. Ovules are attached to the placenta, and that is why arrangement of ovules is called placentation. Ovule is the fertile part. अगर हम इसका longitudinal section काट दें तो in case of angiosperm you will find the most common type of ovule is anatropous, that is inverted ovule, like this. This is the funiculus. Funiculus by which ovules are attached to the placenta. This is the integument. Integument is the protective covering of ovule. And this is the massive nucellus, parenchymatous tissue. Most of the part of ovule is made up of this nucellus, which is equivalent to megasporangium. And in fully developed ovule there is presence of female gametophyte. This female gametophyte will have mostly 7 cells and 8 nucleate structure. Here we will have on the chalazal end there will be presence of antipodal cell, micropylar end, they are the synergids. This is X cell, and inside the center there is presence of central cell. Central cell will contain 2 polar nuclei. This is the structure of fully developed female gametophyte. Actually from the nucellus, first of all ये तो बना नहीं होगा. This entire structure will be produced after megagametogenesis, megasporogenesis. So from the nucellus, first of all, a single cell will differentiate, and that cell will be called archisporial cell. For example, this is the nucellus. So from nucellus, a single cell will differentiate. That single cell will be called archisporium. Archisporium. इस archisporium में there will be covering of callose wall. So it will become megaspore mother cell. Megaspore mother cell will undergo meiosis, producing 4 megaspores. Process is called megasporogenesis. Now, out of these 4 megaspores, 3 will degenerate, 3 megaspore degenerate, and single functional megaspore will survive. Functional megaspore that will develop and will produce the female gametophyte. So rest of the division will occur by mitosis. Till here there is megasporogenesis. After that there will be megagametogenesis. So please note down it. After that we will talk about megagametogenesis. Okay guys, so now this single functional megaspore will undergo megagametogenesis, means megagametophyte development. चाहे आप female gametophyte development बोलो या megagametophyte. तो this single megaspore undergo 3 consecutive cycles of mitosis. First mitosis will occur 2 nuclei, second mitosis will occur 4 nuclei, and third mitosis will occur. There will be formation of 8 nuclei. Out of these 8 nuclei, here you will find 3 nuclei at the chalazal end. This is the chalazal end. Will organize into antipodal cells. Antipodal cells. And 3 nuclei will be organized into egg apparatus. These 2 will remain synergids. This is filiform apparatus जो केमिकल सिक्रीट करेगा पॉलन ट्यूब को अपनी तरफ अट्रैक्ट करने के लिए. Filiform apparatus, and this is the egg cell, and inside the center there will be presence of 2 polar nuclei. This is central cell. So this female gametophyte or megagametophite, this is micropylar end. Micropylar end. That is mostly 7-celled, 8 nucleate structure. Cells are 7, 3 antipodal cells, 1 central cell, 1 X cell, and 2 synergid. Every cell is uninucleate except a central cell which is binucleate. Right? In most of the cases you will find out of 4 megaspores, 3 megaspore degenerate and single functional megaspore develop the female gametophyte. तो दिस फीमेल गैमेटोफाइट आप बोल सकते हो मोनोस्पोरिक, क्योंकि डेवलप किया है एक स्पोर से, मोनोस्पोरिक. दिस इज आल्सो कॉल्ड पॉलीगोनम टाइप, पॉलीगोनम टाइप एम्ब्रियो सैक. In certain cases you will find that these 2 out of 4 megaspore nuclei, 2 megaspore will participate in the female gametophyte development. If 2 megaspores will participate, the development will be called bisporic. And in remaining cases, all 4 megaspores will participate in the development of embryo sac, then it will be called tetrasporic. But the most common embryo sac will be the monosporic, also called polygonum type, that is 7-celled 8 nucleate structure. Now, male and female gametophyte has been produced. The next event will be the act of pollination. Pollination is transfer of pollen from anther to stigma. Pollen grain anther से stigma पे पहुंच जाए तो phenomenon of pollination has taken place. Pollen जब जाएगा तो dehydrated form में जाएगा, dehydrated pollens, right? Dehydrated pollens. अब depending upon destiny of pollens, pollination can be getonogamy, pollination can be genogamy, pollination can be autogamy. Autogamy means you will find here single plant का flower का, flower is bisexual, इसी flower का pollen grain इसी flower के pistil पे पहुंच जाएगा तो process is autogamy, self pollination. Getonogamy you will find कि plants will remain same, for example maize plant, but male and female flowers are different. Male flower is this, female flower is this. So pollen grain from here will reach to the female flower here. Means pollen grain will be transferred to the different flower, but that different flower will be present in the same plant. This is also the same plant, this is also the same plant. तो same plant के जो भी cells हैं they are genetically similar. तो autogamy and getonogamy will not produce new genetic variation in population. Xenogamy. In case of xenogamy, the pollen grain of one plant will be transferred to the pistil of another plant. So xenogamy is only true cross pollination. A pollination agency, you know, so many different types of pollination agency जो pollination कराते हैं, like wind pollination, that will be called anemophily, water pollination, hydrophily. You will find insect pollination, entomophily, bird pollination, ornithophily, so many different types of pollination. Now you will see post pollination events. Pollination के बाद क्या होगा? When pollen grains reach to the stigmatic surface, post pollination events. When pollen grains reach to the stigmatic surface here by the act of pollination, तो the chemicals of stigma and chemicals of pollen grain, इन दोनों का आपस में reaction होगा, and through this reaction pollen grain and pistil will determine whether this pollen grain is compatible pollen grain or incompatible pollen grain. Compatible pollen grain अगर ये recognize हो गया कि this pollen grain is compatible pollen grain, then pollen germination will allowed and pollen tube will grow. Pollen tube जो grow करेगी, that will grow through the style, from the apical portion. Apical portion पे cytoplasm को रोकने के लिए there will be callose plug. तो here will be the pollen germination. Pollen germination के केस में the first event will be hydration, क्योंकि pollen grain जब आया था तो highly dehydrated. जो nutrient और water होगा that will be supplied from apical papilla from the stigma. Nutrient and water. Growth will occur from apical portion. Growth is from apex. Right? अब जब pollen grain style के base तक पहुंचा तो first of all pollen tube will enter into the ovule, then it will enter into the embryo sac. pollen tube entry into ovule is mostly through the micropylar end. Here you will find, here is the ovule, this is the embryo sac, here is this. In most of the cases, pollen tube will enter into the embryo sac through the micropylar end. This is the micropyle. और जिस ये micropylar end से आएगा तो here the process will be called porogamy. Means pollen tube enter through micropyle into the ovule. That is the most common type. Sometimes pollen tube also enters into ovule through chalaza. If pollen tube enters into the chalazal end, then it is called chalazogamy. And pollen tube, if entry occurs through nucellus or funiculus, integument or funiculus, then the process will be called mesogamy. Pollen tube ovule में whether coming from micropylar end or coming from mesogamy or coming from chalazogamy, pollen tube enters into embryo sac always through the micropylar end. सारा pollen tube अगर ovule में इधर से भी आया तो भी वो embryo sac में जाने के लिए इधर से आएगा, यहाँ से भी embryo sac में जाने के लिए इधर से आएगा, यहाँ से भी embryo sac में जाने के लिए इधर से आएगा. तो pollen tube entry into embryo sac always occur through the micropylar end. Here you will find synergids are 2, pollen tube is 1, so one synergid start to degenerate. Pollen tube always enters into the degenerating synergid. इसी synergid में आके pollen tube will burst और इसका जो भी कंटेंट होगा, कंटेंट क्या लाया था, 2 male gametes, 1 vegetative nucleus, ये सारा कंटेंट will be released into the degenerating synergid. सेकेंड synergid को आप बोलोगे healthy synergid, degenerating synergid. तो pollen tube does not enter into the healthy synergid. Pollen tube enters into the degenerating synergid. When pollen tube has reached into the degenerating synergid and contents are discharged, now there will be the act of double fertilization. This double fertilization is the unique property of angiosperm. ये करेक्टर किसी और प्लांट ग्रुप में आपको नहीं मिलेगा. इन डबल फर्टिलाइजेशन यू विल फाइंड देयर आर 2 फर्टिलाइजेशन इवेंट. एक तो चूंकि 2 स्पर्म आए हुए हैं तो 1 फर्टिलाइजेशन को हम बोलेंगे वेजिटेटिव फर्टिलाइजेशन, क्योंकि इससे वेजिटेटिव न्यूट्रिटिव टिश्यू बनता है, वेजिटेटिव फर्टिलाइजेशन. एंड सेकेंड विल बी कॉल्ड जनरेटिव फर्टिलाइजेशन, क्योंकि इससे जनरेटिव टिश्यू बनता है, जनरेटिव फर्टिलाइजेशन. जनरेटिव वेजिटेटिव फर्टिलाइजेशन दैट ट्रिपल फ्यूजन, ये पहले होगा. ड्यूरिंग ट्रिपल फ्यूजन यू विल फाइंड स्पर्म न्यूक्लियस will fuse with the polar nuclei. Polar nuclei before fusion with the sperm nucleus will get fused with each other, producing secondary nucleus. Secondary nucleus. Secondary nucleus is the fused polars. Fused polars. तो ये दोनों आपस में फ्यूज करेंगे तो यहाँ से बनेगा प्राइमरी एंडोस्पर्म न्यूक्लियस पेन, और इस सेल को आप बोलोगे प्राइमरी एंडोस्पर्म सेल. दिस इज ट्रिप्लॉइड, क्योंकि 2 यहाँ से 1 यहाँ से. एंड प्राइमरी एंडोस्पर्म न्यूक्लियस विल डिवाइड टू प्रोड्यूस द एंडोस्पर्म. द चीफ एडवांटेज ऑफ डबल फर्टिलाइजेशन एंजियोस्पर्म इज दिस फॉर्मेशन ऑफ न्यूट्रिटिव एंडोस्पर्म फॉर डेवलपिंग एम्ब्रियो. जो जनरेटिव फर्टिलाइजेशन है दैट विल बी कॉल्ड सिनगैमी. इन केस ऑफ सिनगैमी यू विल फाइंड sperm nucleus fusion will occur with the egg nucleus and formation of zygote. Zygote will develop into embryo. चूँकि ट्रिपल फ्यूजन पहले हुआ था, न्यूट्रिटिव टिश्यू पहले बना होता, इसलिए एम्ब्रियो विल गेट प्रॉपर अमाउंट ऑफ न्यूट्रिशन फॉर इट्स डेवलपमेंट. नाउ कम टू द एंडोस्पर्म. एंडोस्पर्म दैट इज द मोस्ट कॉमन न्यूट्रिटिव टिश्यू फॉर डेवलपिंग एम्ब्रियो इन एंजियोस्पर्म. फ्रेंड्स, एंडोस्पर्म इज आल्सो प्रोड्यूस इन जिम्नोस्पर्म, बट जिम्नोस्पर्म में जो फीमेल प्रोथाइलियल सेल्स हैं फीमेल गैमेटोफाइट की, दैट एक्ट एज एंडोस्पर्म. सो इन केस ऑफ जिम्नोस्पर्म, एंडोस्पर्म इज प्रोड्यूस बिफोर फर्टिलाइजेशन एंड इज हैप्लॉइड, whereas in case of angiosperm you have seen it कि endosperm produced after fertilization and is mostly triploid, but the ploidy level of endosperm is highly variable because in endosperm development high amount of endo reduplication occurs so that the cells also become polyploid. Depending upon the development of endosperm, you will find endosperms are of three types: nuclear endosperm, cellular endosperm and halobial endosperm. न्यूक्लियर एंडोस्पर्म के केस में यू विल फाइंड जो हमारा प्राइमरी एंडोस्पर्म न्यूक्लियस बना है इन केस ऑफ ट्रिपल फ्यूजन, दिस primary endosperm nucleus will undergo repeated free nuclear division without cytokinesis. So large number of nuclei will be produced. These nuclei will constitute nuclear endosperm. बाद में ये सारे nuclei will be shifted to periphery and their cellularization will happen. Means cytoplasm and cell membrane, everything will be synthesized. Right? So that is why it is called nuclear endosperm, because initially there is formation of large number of nuclei. Very common is coconut. In case of coconut, the liquid part is nuclear endosperm and solid part is cellular endosperm, because nuclear finally will become cellular. In case of, this is most common type of endosperm. Cellular endosperm के केस में free nuclear division will be absent. Only after every division of primary endosperm nucleus there will be cytokinesis, and it is mostly restricted to dicots. Halobial is intermediate type, means it has some characteristics of nuclear and cellular both, so and it is mostly restricted in monocots. In case of grains like rice, wheat, whatsoever, the outermost layer of grains are endospermacete. The outermost layer of endosperm is called aileron tissue. This aileron tissue is protein rich and living, right? And that surround the starchy endosperm. Starchy endosperm. Now come to the embryo development. Here you will find embryo development pattern is almost similar in dicot as well as monocot. Embryogeny in dicot is mostly studied in the members of Crucifery family, especially the Arabidopsis thaliana. So here you will find this single celled zygote. This single celled zygote will divide, producing a small apical cell and large basal cell. Apical cell and basal cell. Basal cell will be present towards the micropylar end and apical cell will be present towards the chalazal end. From a basal cell, the division will be in single plane or there will be no division, and apical cell से ही almost पूरा embryo बनेगा. तो first of all there will be formation of this globular pro-embryo. जितने भी developing stage होते हैं उनको हम pro-embryo बोलते हैं. Right? This is the globular stage. After globular stage, this entire structure is suspensor. The first cell of suspensor towards the embryo, that is hypophysis. इस hypophysis से ही root apical meristem, root apex के development में participate करेगा. Root apex development. Rest of the part of embryo will be derived from this region. Now you will find development of two cotyledon in the case of dicots. So on the flanking side of shoot apical meristem, the cells will elongate, and here will be formation of, after globular stage, you will find there will be formation of heart stage या heart shape stage. Here development of cotyledons further continue, and so that plumule will again invaginated, and there is formation of torpedo stage. And finally fully matured embryo will be produced, that is the cotyledonary stage. Embryo will undergo dehydration, mature stage. During mature stage, reserve proteins will be deposited inside the cotyledons and embryo will undergo dormancy. So development of dicot embryo and monocot embryo is almost same, but the final stage will differ. In case of monocot, the final stage will contain single cotyledon. That single cotyledon in case of Graminy family grasses will be called scutellum. अगर आप grasses के embryo का longitudinal section काट दो तो here you will find okay, this is the structure of massive endosperm, right? Here is plumule will be surrounded by coleoptile and radicle will be surrounded by coleorhiza. This is the embryonal axis. This is the embryonal axis. This is the longitudinal section of LS of grain, LS of monocot seed. Monocot seed or embryo. So this is the outermost layer, that is the aileron layer. Aileron layer, that is part of endosperm. This is starchy endosperm. Starchy endosperm. Here coleoptile, that coleoptile is protective covering for plumule. This is single cotyledon. That single cotyledon will be called scutellum. This scutellum is secretory in nature because reserve food is present inside the endosperm. This is embryonal axis. Embryonal axis is called tisellum, जिसके एक एंड पे प्लूमल दूसरे एंड पे रेडिकल. रेडिकल विल रिमेन कवर्ड बाय कोलियोराइजा. Coleorhiza. This is all about embryo and endosperm development. Next, some small articles are present in this embryology, like you will find apomixis. Apomixis means, apo means without, mixis means fusion. अभी तक जो सेक्सुअल रिप्रोडक्शन में आपने देखा, देयर इज वाज गैमेटिक फ्यूजन. गैमेटिक फ्यूजन से ही सीड फॉर्मेशन हो रहा था. दैट प्रोसेस वाज एम्फीमिक्सिस. एम्फी मीन्स बोथ. If reproduction occurs without gametic fusion, then the process will be called apomixis. Apomixis, that is asexual reproduction. Technical term apomixis. Asexual reproduction के लिए आप बोलोगे apomixis. Technical term for sexual reproduction, that is amphimixis. अब asexual reproduction can occur by vegetative method. Vegetative method, जहाँ पे reproductive unit will be the root, stem or leaf having bud, or it can occur by agamospermy. Agamospermy. Agamospermy means, spermus means seed. Seed formation without gametic fusion. Here you will find agamospermy can occur by adventive embryony. Adventive embryony. Adventive embryony में embryo is directly produced from the somatic tissue of ovule. Somatic tissue like nucellus. Nucellus में कोई सेल डायरेक्टली माइटोसिस से डिवाइड करके बना दे एम्ब्रियो. तो दैट प्रोसेस विल बी कॉल्ड एडवेंटिव एम्ब्रियोनी. दिस इज वेरी कॉमन इन सिट्रस, और इसी वजह से सिट्रस में हो जाता है पॉली क्योंकि वन एम्ब्रियो डेवलप फ्रॉम जाइगोट एंड सो मेनी एम्ब्रियो डेवलप फ्रॉम न्यूसेलस. Second type of agamospermy can occur by gametophytic agamospermy, means female gametophyte is first produced. यहाँ के केस में फीमेल गैमेटोफाइट एम्ब्रियो सैक नहीं बनता है, डायरेक्टली न्यूसेलस से एम्ब्रियो बनता है. अब फीमेल गैमेटोफाइट हमें मियासिस के बिना बनाना है, तो विदाउट द एक्ट ऑफ मियासिस फीमेल गैमेटोफाइट कैन बी प्रोड्यूस्ड बाय डिप्लोस्पोरी एंड अपोस्पोरी. डिप्लोस्पोरी एंड अपोस्पोरी, बोथ द प्रोसेसेस आर फॉर्मेशन ऑफ एम्ब्रियो सैक विदाउट मियासिस. अगर मेगास्पोर मदर सेल से विदाउट मियासिस फीमेल गैमेटोफाइट बन जाता है, एम्ब्रियो सैक बन जाए, तो प्रोसेस विल बी कॉल्ड डिप्लोस्पोरी. इफ एम्ब्रियो सैक इज प्रोड्यूस्ड फ्रॉम सेल ऑफ न्यूसेलस विदाउट मियासिस, important is without meiosis, then the process will be called apospory. Process will be called apospory. Ab this embryo sac has produced diploid cells because इस embryo sac के formation में there was no meiosis, तो all the cells will be diploid. Now if इस diploid cell के egg cell से बिना fertilization embryo बन जाए, तो प्रोसेस इज पार्थेनोजेनेसिस. पार्थेनोजेनेसिस, पार्थेनो मीन्स वर्जिन, जेनेसिस मीन्स बर्थ. एग डेवलप एम्ब्रियो विदाउट फर्टिलाइजेशन. इफ एनी अदर सेल प्रोड्यूसेस एम्ब्रियो लाइक एंटीपोडल आर सिनर्जिड, without fertilization, then the process will be called apogamy. So this is all about the apomixis. Please mention it. After that, last part, that is the development of seed. Okay guys, so now I will summarize what is apomixis. That is asexual reproduction. Asexual reproduction, that is reproduction without gametic fusion. Meiosis may be, meiosis may not be. In asexual reproduction, there should be no gametic fusion. That can occur by vegetative method as well as by agamospermy, that is reproduction by seed formation, but that seed formation does not involve meiosis and fertilization. तो बिना meiosis fertilization के seed दो तरीके से आप बना सकते हो, adventive embryonic and gametophytic. In adventive embryonic, the somatic tissue of ovule like nucellus will directly develop embryo, no meiosis, no fertilization. In gametophytic agamospermy, first female gametophyte will be produced without the act of meiosis. Without the act of meiosis, female gametophyte can be developed by two methods. One is diplospory, second is apospory. In diplospory, the embryo sac will develop from the megaspore mother cell without meiosis. So this embryo sac will remain unreduced, that is the diploid. Same is the story in case of nucellus, but the origin cell will be the nucellus. If this embryo sac or any other embryo sac, if egg develop, whether egg is haploid or diploid, if egg develop embryo without the act of fertilization, then the phenomenon will be called parthenogenesis. If embryo develop from any other cell of embryo sac like antipodal or synergid, then the process will be called apogamy. Right? So due to act of apomixis, there will be no variation. Asexual reproduction produces no variation. If there will be no variation, there will be no natural selection, means there will be no evolution. Now if you talk about seed, seed is fertilized, matured, ripened ovule, right? So before this structure, before fertilization it was ovule, after fertilization it will become seed. अब ओव्यूल का ही जो कवरिंग है, दैट इज द इंटेगुमेंट, वो सीड की कवरिंग हो जाएगी, दैट इज द सीड कोट. सीड कोट दैट इज स्पर्मोडर्म. स्पर्मस मीन्स सीड. स्पर्मोडर्म, आउटर यू विल फाइंड टेस्टा और इनर यू विल फाइंड टेगमेन. जो न्यूसेलस था, दिस Nucellus is a nutritive tissue. Either it will be entirely degenerate, और अगर nucellus कुछ बच गया in the seed, then the remnant of nucellus will be called perisperm. perisperm, that is a persistent nucellus, like you will find in black pepper. Persistent nucellus will be called perisperm. In case of embryo sac, there was antipodal cells. Antipodals degenerate. Synergids will also get degenerated. The egg cell has developed the zygote. That zygote develop into embryo. Central cell produces the primary endosperm cell, and primary endosperm produces the endosperm. These are the fate of different component of ovule. These are the before fertilization, these are the after fertilization, right? अब इसके केस में, इन केस ऑफ एम्ब्रियोलॉजी, आपसे क्वेश्चन पूछा जा सकता है, सम न्यूमेरिकल टाइप्स आल्सो, कि लाइक इन द डेवलपमेंट ऑफ 50 in the development of 60 grains of wheat, 60 grains of wheat, how many minimum number of meiosis required. तो कोई भी नंबर ऑफ जाइगोट पूछा जाए, नंबर ऑफ सीड पूछा जाए, नंबर ऑफ ग्रेन पूछा जाए, फार्मूला विल बी n+n/4, वेयर n इज द नंबर ऑफ ग्रेन्स या नंबर ऑफ सीड. फार्मूले का डेरिवेशन इज कि भाई अगर हमें 50 ग्रेन बनाने हैं तो 50 जाइगोट बनाना होगा. फॉर 50 जाइगोट वी विल रिक्वायर 50 एग. ईच एम्ब्रियो सैक विल कंटेन 1 एग, एंड हर एक एम्ब्रियो सैक के फॉर्मेशन में 1 मेगा स्पोर मदर सेल में मियासिस हुआ था, तो 4 मेगा स्पोर बने, 4 में से 3 डीजनरेट, और 1 मेगा स्पोर डेवलप करके एक एम्ब्रियो सैक बनाया था. मीन्स 1 मियासिस विल प्रोड्यूस 1 एम्ब्रियो सैक. तो अगर आपको n एम्ब्रियो सैक चाहिए तो n मियासिस चाहिए फीमेल साइड पे, और पॉलन ग्रेन का नंबर, एक मियासिस से 4 पॉलन ग्रेन बनते थे, तो n पॉलन ग्रेन के लिए n/4 नंबर ऑफ मियासिस. तो यहाँ के केस में यू विल फाइंड कि मिनिमम नंबर ऑफ मियासिस रिक्वायर्ड टू प्रोड्यूस 60 grains विल बी इक्वल टू 60 + 60/4. दैट विल बी योर आंसर. फाइन. तो द आंसर विल बी 60/450. लाइकवाइज आपसे पूछा जाए कि कंसीडर द फॉलोइंग क्रॉस. कंसीडर द following cross. In this cross, female plant is tetraploid and male plant is hexaploid. And then the question goes on saying, find out the ploidy of endosperm. So ploidy of endosperm, you have to see how endosperm is being produced. Endosperm is produced by the act of triple fusion. Triple fusion involves 2 polar nuclei and 1 sperm nucleus that is coming from the pollen grain. अब फीमेल साइड इज 4n, तो जब इस पे मियासिस होगा तो देयर विल बी फॉर्मेशन ऑफ एम्ब्रियो सैक. तो ऑल पोलर न्यूक्लियाई विल बी 2n एंड मेल साइड इज 6n. तो आफ्टर मियासिस पॉलन ग्रेन बनेगा तो स्पर्म विल कंटेन 3n. तो 3n स्पर्म एंड 2n पोलर न्यूक्लियाई, ये तीनों फ्यूज करेंगे. 2n + 2n + 3n स्पर्म, तो टोटल विल बी इक्वल टू 7n एंडोस्पर्म. तो एंडोस्पर्म के लिए आप फार्मूला लिख सकते हो, प्लॉइडी लेवल ऑफ एंडोस्पर्म विल बी इक्वल टू, फीमेल का प्लॉइडी पूरा होगा क्योंकि फीमेल साइड से 2 हैप्लॉइड न्यूक्लियस पार्टिसिपेट करते हैं, और मेल की प्लॉइडी का हाफ. तो प्लॉइडी लेवल ऑफ एंडोस्पर्म विल बी इक्वल टू प्लॉइडी ऑफ फीमेल प्लांट plus half into ploidy of male plant. Ploidy of male plant. Right? So in this case you will find like this, and final structure in case of development you will find fruit. fruit is fertilized, matured, ripened ovary. तो fruit consists of seed and pericarp. Pericarp, that is the fruit wall. Fruit wall. So here in case of fruit formation, sometimes fruit development also occurs without, like seed development occurs without fertilization. If fruit development also occurs without fertilization, phenomenon will be called parthenocarpy. Embryo development होगा तो parthenogenesis, and fruit development without fertilization will be called parthenocarpy. Right? So this is all about sexual reproduction in flowering plant and embryology.